So it's a great pleasure to call on my friend Sava Micic. And when I was first invited by Sigma Tau and by Ashraf here on my left to join in this symposium, it was Munich 2 years ago, and Sava particularly was talking about starting an extremely important study. And of course, it does take a long time to get a good and important study, and it takes a very long time to get the numbers. We all take on board Professor Agarwal's comment about sufficiently powering a study and trying to make it prospective. It's jolly difficult, though, when one is competing with the universally acknowledged fact that the only treatment of male factor fertility is IVF. So we struggle, and it's jolly difficult to get the patients, and I think Sava is to be congratulated on this study, and Ashraf on my left is to be congratulated on making us do it. Sava.
Thank you, Jonathan. Good evening to all of you, Mr. Chairman. I will represent you a few data, some data from this protocol, but at the beginning I have to say that I'm very grateful to head of our andrology laboratory in Belgrade, Prof. Natasha Lalic, who did a great job. It's a great job to do it not only sperm parameters, but biochemical analysis of ejaculate. Which one? This one. The carnitine is a special energy material came into the epididymis directly from the circulation, and through epithelium came to the fluid and to the seminiferous tubules luminal compartment. And active fusion, diffusion is the main way to enter the spermatozoa. What is interesting, it's a high concentration in the epididymal seminal fluid, around 2,000-fold greater than in the serum. You know that is the fundamental, the role is to maturation of spermatozoa, transporting polyunsaturated fatty acids into the mitochondrial apparatus to make a good energy for spermatozoa. And also, Sertoli cell, which is one of the leading and main cells in the spermatogenesis process, also are involved in carnitine's effects. This is the study we started 2.5 years ago, and we tried to evaluate, to emphasize the changes of sperm characteristics. In this speech, we will discuss about the progressive sperm motility. The secondary endpoint was to follow those patients from 3 and 6 months from the beginning of the study, and also to register some adverse events. There is a few adverse events. Around 2% of them have gastric problems during usage of Proxeed Plus, nothing else. It is nothing. This is polycentric, randomized, double-blind, Sava, so many names for one study, but this is very important, and only for those who couldn't make a baby in the last 12 months. We did it 3 types of analysis: classical spermograms, sperm parameters. We will discuss about motility, biochemical analysis of seminal fluid, and we will discuss about carnitine level in the seminal fluid, and also at the end DNA fragmentation, which will be our next time about this problem. This battlefield is always try to be objective, but always with the subjective influence, and we try to be better and better. A lot of use of microscope, Hamilton and others, but not resolve this problem. But experienced technician and lab men could do their personal problems, and they resolve these problems. This is the last revision 6 years ago from the WHO guidelines, that its concentration, percentage of progressive sperm motility. We only change this suggestion to use a strict criteria of only 4% of normal morphology sperms, because it is completely in direction for the IVF and other procedure, not for the everyday lab to do it. Inclusion criteria was a man between third decade and sixth decade of their life, with oligoastenospermia with and without varicocele, and also, as I told you, 12 months we couldn't make a baby. Exclusion criteria is very low, sperm parameter, low concentration, motility, and other endocrine disorders, undescended testis, and other things. Female, younger than 40, with relatively regular menstrual cycles, undercover with a good gynecologist specialist to do that is normal, and only those who are preparing and plan to do IVF procedure in the next 3 or 6 months during our study. Subjective received product according to the randomization. Semen samples was done 3 times at the beginning, at the 3rd month of the therapy and the 6th month of the therapy. There were 125 treated men with the Proxeed Plus at the same sachet, 50 placebo was control, divided into subgroups with or without varicocele. This is the usual and 3 parameters which always discuss with the patient on the meetings, and we will discuss now the 3rd one about, I think, at the beginning of the study, there is 28% of progressive sperm motility. After 3 months of the therapy, there is no significant improvement, but there is a special improvement between 3 and 6 months. We have improvement of progressive sperm motility during the 6 months, which is the 1st point. 2nd point, also there is significant difference in those between 3 and 6 months when we are measuring the progressive sperm motility. It means the duration of therapy is important when we correlated 3 and 6 month therapy. Placebo group hasn't got any statistical significance. We expected that, some improvement, but not more. We want to see what is the impact of some of the groups who will influence this significantly, and we divided into the group with low motility below 10, between 10 and 20%, and over 20%. As you can see, there is a decrease of the group patient with percentage of patients with low motility below 10, and while there is increasing of patients with 20% and more, from 61 to 73.4. This made the significance in this comparison. We made some grouping in the 4 category, over 10, 20, 30, and 40% of progressive sperm motility, trying to find a special group who will be quite good. As you can see, there is 79.4% of people more than 10%. After 6 months, 83%. Some improvement, but this is not statistically significant. On the contrary, on the group more than 20%, we have at the beginning 61.5 and improved to 73.4. This comparison is very significant. The next group, 3rd group, those who has more than 30% progressive sperm motility, from 44.4 to 51.1, also improvement, but not statistically significant. And the last one, the best group, more than 40% at the beginning, very low number, at the end after 6 month treatment, a very good number, and this is also significant improvement. The second thing is to try to measure seminal carnitine before, during, meaning at 3 months and at 6 months. And we got the result that there is, got results, the significant improvement in concentration. It's quite high at the beginning, but it's much more higher at the 6 month treatment, and this difference is quite good. After that, we make some division between those who have relatively high, over 700 micromole per liter, and those who has lower, below 700 milliliter of carnitine in seminal plasma. And this is a very interesting thing. As you can see on the first slide, there is improvement in those with a low concentration, relatively low concentration, below 700 micromoles. Also, much more improvement in the group with higher, more than 700 micromole of carnitine. But at the beginning, the result was the progressive sperm motility is very correlated to the concentration. Those at the beginning with the low has a lower progressive sperm motility at the beginning as compared to the group with a higher concentration. And there is also difference, significant difference between the 3 months in the lower group and 3 months at the higher group of seminal carnitine concentration. The question is, what is the optimal cutoff for carnitine, improvement of carnitine? We try to find it, and we find it that is about improvement or increase more than 7.7%. It will be quite a good cutoff for those patients. And we divided according to this to 2 groups. As you can see on the first group, yes. There is no improvement in the group with the increase of sperm carnitine is lower, below this cutoff, but it is significantly improved in the group with a higher increase, more than 7.7%. Also, there is significant difference between, at the end of therapy, between the lower group and the higher group of increase of seminal carnitine. This correlation is also interesting. Also, we have to divide in previous 4 groups, 10, 20, 30, and 40%, and try to find is any difference in groups with a lower increase and with high increase. There is no statistical difference in each, with 10%, more than 20%, and also for those with 30 or 40%. There is no significant increase in number of patients with progressive sperm motility for 10, 20, 30, or 40%. And in conclusion, we can say that we found progressive motility was much better after the treatment. We found that the group of progressive motility more than 20 and 1 to 40 is increased significantly after 6-month treatment, not only after 3 months of treatment. On the other hand, it is important to find that carnitine affects progressive sperm motility. We found that carnitine is greater in seminal plasma, the greater is the progressive sperm motility. Also, this cutoff and more than 7% increase of seminal carnitine influence our expectation that the progressive motility be more than 10% with very high confidence. Also, we divided those groups with low and over 700 micromole of seminal carnitine is better at the beginning, better progressive sperm motility at the beginning, those with high, and also at the end of the therapy in comparison with those who had lower seminal carnitine. And also, there was significant increase in both groups. We can use it in both groups, and we, on the other hand, try to make some mathematical differentiation. And then we can say that the study confirms that the high seminal concentration carnitine plays an important role and confirms that the therapeutic usage of carnitine is effective in improvement of progressive sperm motility during the study. And this is always our aim to do that and to use that. Thank you.